Hi, good evening, everyone. So Sarah grew up in the, well, her love for nature began at a young age when she was growing up in the Chatsworth area, where she attended the Holland Chatsworth Central School, and then went to high school at the OSCVI in Owen Sound, where she was, I think, influenced a great deal by Jim Sebastian. And with a desire to explore the world's oceans, she moved to Halifax in 2013 to study marine biology at Dalhousie University and researched longfin pilot whales and cultivated her skills as a wildlife photographer on the ocean. And she graduated with a degree in marine biology and a certificate in animal behavior in 2017. Continuing her newfound love for oceans, she moved to Vancouver the same year and began researching Gentoo penguins with the University of British Columbia, traveling to the Falkland Islands in the Southwest Atlantic Ocean. And she spent total of 9 weeks over spring and fall, observing the Gentoo penguin colony. And she graduated with her Master's of Science in Zoology in 2020. And at the same time, she also won the Warner and Hildegard Hess Research Award for her master's paper. And after finishing her master's, Sarah and her partner, Spencer Turbit, moved back to Owen Sound to be near family, and for Sarah to pursue painting for a year before deciding on her future career line. After being invited to become a member of the Artists for Conservation, Sarah made the leap from science to full-time art in 2021. And Sarah and Spencer are new parents with Sidon, born this past September. Please welcome Sarah Turbit McComb.
Yes. I'll put y'all to sleep. Okay, awesome. So yes, I am going to be presenting today for how to catch penguins. So we're going to talk about some of my field work and how I combine science and artistry through research in the winter foraging of penguins. And we're going to share their story through wildlife art. Is that better? Yeah, can you guys hear me okay? That's better. All right, here we go. I might. Okay, there we go. Now the slides are bigger. Okay, so we're going to do a bit of an adventure highlight for today. So we'll give you an outline of what to expect. So we've already kind of done a brief who am I, but we have a couple quick pictures. Then we're going to introduce the Falkland Islands, talk a bit about why Gentoo penguins were chosen to do my research. And then we're going to get into the details of the field work. So how we catch penguins and work with them. And then my main research focus is around the idea of individual qualities. So how an individual animal outperforms others of its species. So we're going to talk about measures of their condition, their body condition, and how this affects their diving efficiency, so their ability to catch prey when they're going into the water, and also how this relates to their breeding status, so how successful they are at reproducing. And then we're going to go into painting wildlife. So we already kind of got a bit of an introduction to me, but I was drawing from a very young age and obsessed with the ocean. So this is actually at our trailer in Lion's Head, and we're coloring there. And then this is a few clips of adventures from out east. So we went there and I lived there for 5 years, worked with seals, sea turtles. We did a lot of whale stuff. And eventually this brings me to living in Vancouver and working with penguins. And then now full circle, we're back here with all of these adventures and we're trying to share it with creating wildlife art so we can tell these stories and also the stories of other local animals. So diving into the cold, how variation in winter foraging behavior relates to oxygen stores in penguins. So Gentoo penguins, we're going to go over some quick facts just to get you a bit more familiar with them as a species. And they can dive deeper than 200 meters. They are the fastest swimming penguin. They are just shy of 1 meter in height, as we knew from our trivia. They're not migratory, so they kind of stay in the Falkland Islands all year round. and they are long-lived. They live over 20 years. And ideally, they can have 2 chicks per year. So they're a little unlike emperor penguins and other species like that. So they can raise 2 a year. So there are 18 species of penguin in the world, currently still with us. This would have been a good trivia question for the calendars. And they're divided into 6 genera. So of this, most of them are endangered or threatened to some degree, as a lot of seabirds are really struggling in today's environment. But one of the least concerned species is actually the Gentoo penguins. So one reason for this is because Gentoo penguins are generalist foragers, so they can actually eat a wide variety of prey in the ocean. So there's different krill, fish, and shrimp, and their foraging behavior is adapted for a wide range of species that they can encounter. So they're really adaptable to the local conditions. So one thing that makes them unique, though, and why we targeted the Falkland Islands for our research, is because they're actually a separate genetic population. They're not officially recognized yet, but they don't interbreed with the other penguins of Antarctica, largely because there's a genetic barrier of the polar front. So the penguins don't cross this line of the ocean from the currents that circle Antarctica. And the amount of adult penguins that are participating in breeding vary drastically from year to year. So you have these huge spikes and drops in breeding pairs, and it's really unknown environmentally why this happens. So this is data from Falkland Conservation, one of the organizations that I'm now supporting with my artwork. And yeah, so we were kind of looking into this as an initial like seed of thought of why we wanted to check out this animal. So one other thing we're going to do for background of penguins is talk about their annual cycle. So it's color coded because it is opposite to us. So when we talk about summer months, we're talking about December and January, unlike up here. So right about now, their Gentoo penguins are laying eggs, and they're sitting on eggs, incubating them. Then they brood for about 40 days, and it takes about 14 weeks for their chicks to age up into adults. And then as an entire colony, they all molt for 2 to 3 weeks. This is the most energetically expensive time to be a penguin, because unlike birds that can just drop and regrow feathers kind of on the go, on the fly, if you will, then penguins have to do it all at once. So they molt all their feathers, and they can't go into the water because they're not waterproof while this process is happening. So after ideally raising kids and shedding all your feathers, so you have a brand new coat on for winter, they really have a goal of increasing their body condition so that they're able to breed on time for the next year. So, and then the cycle just repeats each year. So now we're going to get into the question of individual quality. So the definition of this is a collection of phenotypes that are positively correlating with the animal's fitness. Fitness being the number of offspring that they can reproduce, so their genes are carried on. So one of the main questions in ecology is, how are high quality individuals outperforming others? So while we can't assess individual condition directly, individual quality, we can use some proxies for their condition. So this can be their body health, their fat reserves, nutritional state, and also their physiological performance, such as their ability to dive. And all of these are going to closely relate with their foraging success. This now brings us to the Falkland Islands. So we're going to go to a journey just off the coast of South America. And this is the one town in the entire country. So it's about 3,000 people, and otherwise people are just kind of scattered throughout the islands in remote camps, they call them. So it's just a cluster of buildings, and there's a small plane that drops off supplies 2 or 3 times a year, depending on which island you locate off of this. This is what the small plane looks like. So we went out there with a team of 4 initially. And the Falkland Islands is a really unique mix of intense wildlife and also a lot of farming. So there's sheep, there's some horses that you'll encounter with the penguins, which is fun. So this is what the landscape of the islands look like. So it's so windy there that trees can't grow. It's just constant. Like I've never been in a more windy environment in my life. And there's lots of different wildlife that call this place home. If you ever get a chance to go, it's a really amazing place. And you can get so close to animals and they don't even really react to your presence. It's wild just because there's not that many people, especially when you go off of these more unique islands. So there's lots of different species of marine mammals and avian species as well. And we're going to get to know a few of these when we talk back about sharing their stories through artwork. So the Falkland Islands is really unique because it's one of the only places in the world where you can see 5 different species of penguins. So we have gentoo penguins, we have our king penguin, which is the 2nd largest species, and they live on sandy beaches. Then we have our Magellanic penguin, which is the resident burrowing penguin. So it's got a big mouthful of grass taking back to line its burrow. Then we have our cliffside penguins, which are the macaroni penguin, which is a bit more rare, and the southern rockhopper penguin. So what's really interesting is even though there's 5 species and they're all penguins, they all have very distinct nesting environments that they're occupying, with the gentoo penguins being inland in bushes. So in April, we went down as a team of 4 for 3 weeks. And this was with Marie, my supervisor from UBC, and Dr. Glenn Crossan. He was our local expert of penguins that taught me how to take blood samples and handle them and all this. So we were all there. And then in October, when I went down for 6 weeks, it was just me and another master's student. And this is an example of how you catch a penguin. Not quite, it gets away here. But anyway, for obvious reasons, how we catch penguins, we are not able to do it in the ocean as the sea lions have enough trouble with it. Then on the beach, you have these giant rocks, which would be hazardous for both the penguins and ourselves. Then we have this lovely grass habitat, which would be very easy for us to run across. But 2 reasons: gentoo penguins actually can run faster than humans, surprisingly. And additionally, that's the habitat of the Magellanic penguins. It's where their burrows are. So for our ankles and their homes, we are very careful when we tread in this region. So there's this bush called the diddledy bush, and we have longer legs. So that's how we are able to net the penguins. And then we are working with them for just a few minutes before they're released again. And also the colony is up their path from the ocean, and we don't want to disturb their nesting sites. So we stay far away from where they're actually nesting. Then we take a quick blood sample and measurements. And I attached 2 different types of devices to the back of penguin. So this is a stencil just to make sure that any glue drips don't touch their fur. But we have a Argos satellite tag, which is going to send its location. And we also have a time depth recorder. This records the pressure for the depth that the penguin is at every 1 second for a month, which is a lot of data points. So then you can kind of see the method of folding up the tape around different layers of its feather. And then on the top one, we've super glued the seams. So that's all that's keeping these tags on, and they fall off on their own, or we could remove them if we find them. And off they go to collect all our data for us. So the goal initially for my work was to combine their movement data with dive profiles. So this would allow us to detect where in space they were just doing exploratory dives versus where they were actually finding prey and foraging. So this was the original goal of my thesis. However, we ran into a few difficulties. So this is a graph of the bathymetry, which is the depth of water across the Falkland Islands. And you can see a few dots start to appear on the screen here. These are 5 penguins wearing my tags. So from April to about September was the goal. I woke up every morning and checked where all my penguins were. We tagged 66 animals, and you might be wondering why there's only 5 here if 66 were tagged. A lot of the penguins started dropping off around late May. So slowly we watched our project disappear into the abyss. And these are the last 5 penguins who lasted the longest. So you can see that a lot of them are staying pretty much on the shelf and within 100 meters depth of the ocean. And when you get into late summer, you're seeing a lot more penguins are being forced to go a little further to try and find food. This is because the prey resources kind of get a little sparse in the later months of winter. So a lot of the penguins are making day trips, but as you see, a few of them are really staying out for quite a few days at a time. So now we're into August and there's 3 penguins and 2 penguins and 1 penguin. So this is about a month before I was to go for my field work. And basically, it was a bit of a discouraging point in our journey of getting our master's degree because we really didn't know if we had any data at this point. It wasn't enough movement data to really base a project off of. So we were really just hoping that some of the time depth recorders, which was a smaller tag without an antenna, would last. So if you spot a number on a penguin, just yell it out. We have an animal dye that we use for their front. So you're looking for numbers on their belly. All right, the video's looping. Did anybody see one? Yes, so 59 and 48. 48's just walking off the screen on the bottom right. And I think 59 is going to jump up on a rock to the left. Yeah. So while we were searching for penguins, we also had the anxiety of watching the sea lions hunt, as I already kind of showed this clip once before. And although this sea lion was not successful, many were, and it was very adrenaline induced because we weren't sure if those were our penguins that were getting caught. But luckily, we did find time depth recorders. So it was a huge relief to spot them in the colony. But if the penguins are in the colony where we cannot capture them, we have to wait until that animal leaves to go fishing and then returns to try and catch it again. So it was many days of effort to try and collect a few tags. So out of 66, we were able to recollect 21, which was a very, very great day. And we knew which penguins still had tags, and it was just like, you know, a game of chase to try and get all of them off them. So these are what the time depth profiles look like. So it's from April until October that they were collecting data for us. And similar to how the movement data showed, the penguins are staying a bit closer to shore in shallower waters. And then they're doing really deep dives later in winter when food becomes more scarce. So back to our question, how are high quality individuals outperforming others? We're going to talk a little bit about how animals are able to hold their breath. And this is the same for humans as well. So it's an important constraint on diving performance and foraging. So we can think about it as if we were trying to collect rings off a bottom of a pool. If you can only get one ring every time you go to the bottom, there's different ways that impacts how many rings you're able to get. On the other hand, how long you have to catch your breath at the surface before you're able to dive again also will impact how successful you are when it's a bit of an endurance run. So the ways you can store oxygen is in your lungs. You can take a deep breath before you dive down. But we also have oxygen carrying proteins built into us. So we have hemoglobin in our blood, and we also have myoglobin in our muscles. So myoglobin is harder to measure when our objective is to leave the penguins as undisturbed as possible. So we took quick blood samples and did a lot of our measurements from there. So of that, we have our indices of oxygen storage as their body mass. And we also have a hematocrit, which is the packed red blood cell volume, and hemoglobin, which is the amount of oxygen binding protein in the blood. So our first question we're going to ask is, do the condition of oxygen stores affect individual diving efficiency? So out of the 40 days of pre-breeding dives based on the life cycle of red blood cells in penguins, we have collected over 100,000 dives that we are using in our research. And you can see that there's foraging clusters at 60 meters depth and also at around 150 meters depth. So now we're going to talk about inferring behavior through our dive metrics. So we really only have a very simple data. We have the time and the depth pressure, but with it, you're able to create these profiles of their dives. So there's some key points that we're able to pull data from, like the maximum depth of a dive, also their bottom time, which is their most likely place to find prey. And we have their total dive duration, including their time to travel down and back up. And also we have their post-dive surface interval where the penguins are catching their breath at the surface. So when you divide their bottom time by their total length of their dive cycle, you get their dive efficiency, which is how a penguin can maximize the amount of time it can spend with prey. I'm going to breeze over generalized additive mixed modeling, but basically you have linear models, which is X, Y, then you have generalized mixed models, then you have generalized additive mixed models, and then this is where we get to. So if anybody wants to go into the statistics of how we did our research, I am happy to discuss it, but I won't go into too much detail with this part here. Basically, all this comes down to is that when you do a selection criteria for it, you can see that hemoglobin, hematocrit, and mass show up as being the most relevant as they're on the top of the expression graph. And they have the lowest difference in AIC. In more visual terms, basically in hemoglobin as the top, then we have hematocrit, then we have body mass. You can see that their dive efficiency is relatively similar on all three graphs from the first 0 to 100 meters depth of diving. But when we look into deeper dives, we see a lot of variation in the different indices of oxygen storage. So to talk about hemoglobin, for really shallow dives at 50 meters, there's not that much difference if you have low hemoglobin or high hemoglobin in your diving efficiency. But it becomes a lot more drastic if you have low hemoglobin, you're a very inefficient diver at 150 meters compared to penguins with high hemoglobin, which are still up in the green. Interestingly, for hematocrit, we found that there's a bit of a curve. So penguins with really low hematocrit and penguins with really high hematocrit didn't do as best at 150 meters as penguins with a medium amount of hematocrit. So this is really interesting because your density of red blood cells also impacts the viscosity of your blood, how thick it is to move. So it makes sense that there would be an optimum level. And for mass, there was a slight effect, but not as strong. So if we focus into the dives deeper than 140 meters, we get a drastic change in dive efficiency for hemoglobin. And we wanted to figure out why this was the case and look into it a little bit more. So if you remember, we were using dive efficiency as bottom time over their total dive cycle. So we wanted to first look into their surface recovery time. So are penguins with higher or lower hemoglobin taking longer or shorter to catch their breath once they're back at the surface? And the answer to this was not so much. It takes them about the same amount of time to catch their breath and dive back into another dive. Where we did find a difference is in the bottom time. So the amount of time that they're actually able to spend underwater where the prey is. So even though it's statistically significant in terms of numbers, a question you always want to ask in science is it ecologically significant? So we found that it's an average of about 16 seconds that they can spend at the bottom time of their dives. which is about 22%. So we think that this is highly likely to impact their foraging success. And we can also see this has an influence on their behavior, as penguins with high hemoglobin dove beyond 140 meters, about 3 times as likely. And penguins dove about twice as likely beyond 70 meters. So because we know that food is becoming scarce inshore, this really gives penguins a big advantage when they're having to go far out and dive deeper during this time period right before breeding, when they're trying to collect their energy reserves. And their individual diet reflects their diving behavior. So these are from plankton and bottom trawl surveys. And it kind of shows you where different types of species are and the different energetic density that they could be finding there. So there's a lot more options for prey available if you're able to dive these deeper depths. So one thing we're finding with our penguins is that you can have generalist individuals or specialist individuals. Such as humans, we're all able to eat a wide variety of stuff, but you have your favorite dishes that you cook over and over again at your house. So penguins are similar to that, and they have individual specialization of what they like to eat. And this is relevant because individuals can actually subdivide a population's niche, which means that if there's a pressure on their system, such as from a fishery, it doesn't affect every individual the same. So individuals that have become specialized to hunt at 70 meters and don't necessarily need a lot of hemoglobin would be impacted a lot differently than penguins who are choosing to really specialize in these deeper environments and hunt there. And what's interesting is you see both these types of foraging behavior from the same colony. So to summarize our first question, lower hemoglobin does reduce dive efficiency at deeper depths. And it's important to verify that we see these indices of condition are actually causing impacts. We also noted that an optimum hematocrit was around 52% from our data, and suboptimal oxygen stores hindered their ability to forage at depth. Okay, so on to our second question here is foraging effort influenced by condition and breeding status? So does this actually have an impact on their penguins' ability to reproduce? So if we go back to the annual cycle here, in the winter, they're really trying to increase their conditions so that they can lay their eggs on time. And we see this a lot in a lot of different bird species, that if they're in poor condition, they can delay breeding so that they have more time to increase their energy reserves, or they might skip a breeding season altogether. And if you remember the chart that we showed at the beginning of the really drastic increases and decreases in the number of breeding pairs, this can really affect that. So we wanted to be able to link foraging to differences in the breeding behavior of our penguins. So we did this through colony observations. We spent many hours looking at our penguins and noting different breeding behavior and their lay date. So we were really watching to see if penguins when they were laying their eggs. So this is their calling behavior. They also have aggressive displays to defend their nest territory. Pairs bow to each other is a common mating ritual that they do to reinforce their partnership. And there's also nest building behavior. So this was really funny watching this happen, because you can see the nest has actually gone all the way over this penguin's back. So there's not really an off switch for some of these behaviors, but they just continuously steal sticks from each other. And this is actually penguin number 58, which was my dad's penguin. All my family members got to have a hand in naming some of them. And this is actually him looking at his egg for the first time. So we watched him come back to the colony that morning. There was no egg the day before. And this is him looking and realizing that there was an egg in the nest when his partner stand it up. And ideally, they lay 2 eggs about 3 days apart from each other. So this was the maps that we would create of our nesting locations. So we used these as a guide so that we could easily kind of compare our notes to where we saw the penguins today between me and the other observer, Joanna. And basically you can see numbers 50, 56, and 49 here. And we looked at their ability to participate in breeding. So whether it meant that they were a non-breeding or breeding individual displaying these signs. And we didn't really find a significant difference there, but they could forage differently. So we had metrics of foraging effort. So how much time that they needed to spend. This was in trip duration, time at sea, and their diving effort. And we did a principal component analysis with this data. So essentially for the principal component analysis, the first kind of percentage of explained variation in our data is basically says on the bottom axis there that penguins with longer trip durations spent more time at sea, but had less dive effort. And interestingly, on our second component of our analysis, more time at sea and diving effort increases, but it's independent of variations in trip duration. So the total time a penguin spends at sea. I'm going to skip the exact details of the principal component analysis, but basically all our penguins who were early laying penguins had the least amount of time spent at sea, and they had shorter trip durations. So is foraging effort influenced by condition and breeding status? Efficient foragers are spending less time at sea and were able to lay eggs earlier. And time on land is essential to prepare socially for breeding. So there's pair bonding, nest building, and nest defense are all important behaviors that penguins want to be on land for if they're planning on raising chicks nice and early. So to kind of summarize all this research, foraging is essential for survival, reproduction, and population dynamics. And it's a priority for seabird research to understand what drives their foraging behavior, because when their populations aren't doing very well, with a lot of penguin species are in this situation, we're really at a loss for how you can describe what is going to help them the most efficiently. So it's got a lot of conservation implications to understand this. And we looked at the importance of individual oxygen stores for this. We also looked at the monitoring methods that best reflect their overall condition. And we have a lot of support for using hemoglobin as a condition. And we have caution using hematocrit because higher hematocrit doesn't necessarily mean that they're better divers because of the complications of viscosity issues. So there's an optimal level for hematocrit, and that's really hard to tell in field, especially with smaller sample sizes. So there's more support for hemoglobin as a more accurate measure of how a penguin is going to be able to perform. And we're focusing on diving performance here, but it can also be extrapolated to other migratory animals, especially animals in flight, other bird species that can, this will be a condition of their performance as well. So variation in diving performance affects their prey selection and things like their interspecific competition. So how individuals are competing with each other. So if you're able to dive physiologically to these deeper areas, there could be a lot more prey there because you're not competing with the rest of your colony. So specializing in an individual can be quite beneficial because there's less competition. And additionally, the impact of fisheries could differ between individuals based on what prey they're targeting. And we do see population declines of gentoo penguins in other islands that are away from Antarctica as well. So it's something that we really want to monitor with the Falkland population. And gentoo penguins can help us understand the reproductive trade-offs that animals must consider. So whether or not to skip a year for breeding is a big question in biology. Okay, so that's the end of the research part of our talk. This is the team. So Dr. Marie and Glenn were amazing to learn from, and there's tons of other people who were involved in logistics, and especially Falkland Conservation provided us with a ton of support when we were on the ground. So now we're going to talk a bit about sharing their story of these animals through artwork. So these are gentoo penguins. And this was an oil painting I did in 2018 during my master's. of Gentoo penguins from that video. I kind of composed a few of the penguins from here. So this was the only painting I created when I was doing my master's. I'd always kind of been a hobbyist painter. I painted a lot when I was younger. I took art classes with Sebesta at OSCVI, but you get really busy in a master's degree, as you could imagine. And basically, I kind of left art for a while, other than as a hobby, until I came across the Silent Skies mural at a conference. So this was at the Ornithological Congress in Vancouver in August 2018. And there's certain moments where you have an experience and it's like a pinpoint, you can pinpoint a moment that was just pivotal in everything that you thought you could do in the world. And seeing this mural was it for me. So there is a 678 endangered species individually hand painted as part of this mural. It was an effort from 170 artists across 15 countries to produce all of this artwork. And it's just amazing walking along it and being able to visually see all the endangered bird species in one place. It was so impactful and it was just like an amazing thing. So while at this conference, they had members of Artists for Conservation live painting at the event, which is where I met David Kittler. And a year from meeting him, I was invited to take a weekend painting class with him when he was traveling to Vancouver. So I picked a painting from a clip from my research, and I eventually created this penguin painting of the rockhopper penguins. So I started it in the class in late 2018, and then I took until 2020 to finish it when I finished my master's thesis, and then we were moving back here. And this is the inspiration for that painting. I can also mute the audio if it's annoying. Yeah, so you can hear wind and you can also hear the chattering of the rockhopper penguin calls. They make that really high pitched screeching noise. That's what they sound like when they call. All the penguins have very different vocalizations, which is pretty fun. There we go. Yeah, so basically after running into Artists for Conservation at this event, I became obsessed with the idea of joining them. I applied twice, and in 2019, I got accepted as an associate member, which was really great. And in 21, I had a painting of mine, Astral Seas, get selected for the 200 paintings in their exhibit. So as an associate member artist, you're allowed to submit artwork. And then their signature members are limited to 500 members. So Astral Seas was created from a challenge I did to raise awareness for the different species of cetaceans. So I did a 31-day challenge in January of 2021 with 15% of the proceeds going to the Society for Marine Mammalogy Conservation Fund. This is the same society I planned their art exhibit for their conference that was down in Florida in August this past summer. And yeah, so we were able to paint 31 species of cetaceans. You would never know it, but a lot of these animals are actually painted from stranded specimens. So I used a lot of my networking with other marine rehabilitation organizations and stranding networks to collect photographs of animals. And you can cover a lot with paint. So you can really patch an animal back together and still get the proportions that are accurate to the species, which we really thought was important from a naturalist perspective. So moving back to the Falkland Islands, I also have captured the southern giant petrel. These are gliding birds really closely related to the albatross, and they have a huge, massive wingspan, and they just coast on the intense winds in the Falkland Islands. We also have a South American fur seal. I actually decided to keep this painting because it's the first one I created when I came out of my master's thesis, and it's in front of us here. So these are some other inspirational moments that I hope to capture soon. And all of the work from this is supporting Falkland Conservation, the organization that I partnered with for my research, which is fun. So you can hear their calls kind of over the wind. The Gentoo penguins sound a lot nicer than the rockhopper penguins. They're a little less shrill. In the Falkland Islands, they also have black-necked swans, which I thought were particularly beautiful. So there was one single breeding pair on Pebble Island, the island that I was there for my research. And we only saw them about 4 days out of the 6 weeks that we were there on their interior lake here. I also have to mention the striated caracara. This is one of the most, how do I say it? Curious and annoying animals I've ever encountered. They would hover about a meter above us when we were looking through binoculars, trying to look for our penguins coming to shore, which, as you saw, is an activity that creates, demands a lot of focus. And on the left here, you can see they're actually picking apart the rubber of our rental car. So they're very fun. But they are beautiful animals and very curious and unafraid of humans. So they would come very, very close and always wanted to watch what we were doing. We also had southern sea lions, which were really fun. Such a contrast as a biologist to see them when they're hunting our penguins at our beach. And we're like, no, get away, penguin. Like, we're cheering for the penguins. And then this was an area where we could look over the cliff face, and about 10 feet down were these wild sea lions nursing and in their colony. And it's just crazy to think like, you're the one that's killing our penguins. But, you know, your pup's cute. So I don't know. And towards the end of our visit, in their great anticipation of the many penguin eggs that were going to be arriving, we had the Falkland skua, which, if you note, it's the main bird avian predator from Happy Feet. And although on Pebble Island, king penguins do not nest there, there's not really a beach that they have a preference for. We saw one. And it was crazy. We thought somebody was standing on that sandbar that you can see in the distance. So we pulled our car over when we were off-roading to our field site. And this king penguin walked right up to us, just so curious. And we probably spent about half an hour with them. And we're just sitting on the rocks, and he comes right up to you and then is going on his merry little way. King penguins are very massive. So it's like a small child. It's crazy. So those are some of the moments that I hope to turn into paintings eventually when time permits. One thing I did this year is to try and diversify my skills as a wildlife artist. Is I did a diversity of life collection. So last January, all these animals were voted on social media, particularly Instagram and LinkedIn. And we made an entire collection. The goal was to get to 31 again, but these were a bit more detailed. So we ended up with just about 20 paintings, I think. And the proceeds from all these are going to be donated to Wildlife Preservation Canada. And I was invited to be one of the signature members of Artists for Conservation, which was a life goal of mine, which is very exciting, just this past summer. And I had 2 artworks accepted into their art book. It's available on their website. We can send the link out somehow. And my painting was selected to be the cover of their calendar, which is still something that I think is crazy. There's so many talented artists featured within the calendar. If you're lucky enough to get one of the 10 that Jeff sent from Artists for Conservation for us tonight, which was very generous of him. And yeah, there's like Guy Harvey and a whole bunch of really amazing artists, including David Kittler, who I took the class from when it was 2018. I also have a YouTube channel, and we're 8 subscribers away from hitting 1,000, which is crazy. So I started this last January, and there is painting tutorials to show how each of these paintings were created. So I recorded a time lapse of me painting, and I talk a little bit about different choices that you're making as an artist to create these realistic wildlife art pieces. And I also talk a little bit about the animals that were chosen for them. So it's meant to be a platform to spread awareness about lesser known species, as well as teach how to create wildlife artwork as well. And as you know, I have a studio just down the street from us. I'm just behind Mudtown Cafe, and I have a baby, so I'm not always there now. But if anybody wants to come and say hello and see what we're doing in the studio, I would love it for you to stop by. And for anybody who didn't see the penguins as they popped up before, here is a repeating loop so you can test your skills and try and find them now. And thank you all for listening. Yeah, we can have questions. Yeah.
So we'd like to invite questions from the audience. Yeah. So fire away. So the question is data transferable to ducks? Yeah, comparable.
Yeah. I'm not sure what you mean by the data itself would be transferable, but the methods, absolutely. So if we had time depth recorders, they would record from, as I'm saying this, they're saltwater activated, but I'm sure there's other devices that would record just with the pressure change of fresh water. Yeah, absolutely. So hemoglobin would be a great indice of how the different ducks would be able to hold their breath. Yep.
Yes. Yeah. So the question is human activity noted to affect penguin behavior or penguin outcomes? Yeah.
Yeah, so there are many different fisheries that could potentially be causing an impact, but it's hard to know that for certain. And we had repeated years of study where we had this diving behavior, and we could see that, oh, a fishery came in for this species. All of a sudden, all these penguins are avoiding, you know, depths of that level where they're found at. That would be a bit more telling for something like that. But without years of repeated data that are monitoring it before and after the fisheries were put in place, it's really kind of hard to get those finite details apart, because there's so many different variables that can, you know, influence the cause and effect of how different stuff is happening.
Yes. So we're looking for, the question is the iron source to promote hemoglobin production in the penguin.
Yeah, so I don't have the iron concentration of their different prey species off the top of my head, but it would be from different prey species. I think squid is lesser high in iron. I think other fish species are high in minerals, but there are different trace minerals in all of these, and they get it all from their diet. So
John at the back. So the question is, are they strictly carnivores or?
yeah, they are strictly carnivores. There's some phytoplankton that might be consequential as they're foraging other things that could get filtered in, but I think it would be such a trace amount. They're really after larger prey items that they're able to swallow. And one thing I will note is from our introduction slide, you notice that the penguin is actually drinking water from a puddle. Penguins get most of their water from prey as well, and they have very sophisticated salt glands to filter it out, but we did see them drinking water occasionally. So the fact would be that penguins get water from their food, but there's some other things that can happen, like we see them drinking water occasionally. So there might be some stuff that they're getting, but they don't target anything that is plant-based in the ocean.
I'll just throw one question in. Sarah, my head is still spinning from the mathematical formula and the graphs. Did you guys have any help from the math department?
No, this is just all me. Our lab is the Statistical Ecology Research Lab, so it's mostly modeling. This is how they trick you with science. People say they want to be a marine biologist to avoid a desk job, and as you saw, it's like 3 weeks and 6 weeks were spent in the field. The other 3 years were spent doing that formula. So it's a big con of biology. Biology was actually my worst mark in high school, which is funny. So.
Question at the back, yes. Question is penguin bearings.
That is a great question. Navigation in animal is basically, as far as we know, and talking in a very broad sense, they can use light cues, they can use the position of the sun and other environmental impacts, but how animals migrate and travel is so complicated as a topic that it would be fascinating and a whole presentation on its own to really try and piece that apart. One big thing that I had to learn was just the timing of breeding. So how did we know when to go down for our 6 weeks to try and catch when they were going to lay their eggs? So the amount of books that go into deciding environmentally the timing of breeding, because you have to time up when they're going to start physiologically producing their egg. And that has to be like, depending on the species, it's like 100 days before it's actually going to be fertilized and then it's going to be laid. So all of these events are put into motion really far advance of knowing when there's going to be good prey conditions. So timing of breeding in birds in general is a very complicated thing when you actually think about it to try and tease apart. So, yeah.
Okay. Ryan at the back. So the question is that reading through or? No. Okay, they've got it on.
Yeah, so climate change is affecting different species of penguins, especially because as Antarctica is warming, it's creating more nesting habitat that's not covered in snow. And that is better for gentoo penguins, which is why their population in general is of least concern and increasing. There's more habitat for them to create nests in Antarctica. And unlike on the Falklands, they don't use sticks, they use pebbles. So gentoo penguins are the species that classically will propose to their mates with rocks. They bring them pebbles to create nests. So yeah, but for a lot of penguin species, for prey conditions, for the cascade of effects that are happening in the ocean, it is being very detrimental to penguins as it is a lot of various seabirds, because the oceans and trying to find where prey are is very complicated when your prey is swimming and you are not for a lot of other seabirds. Penguins are, but the ocean's still a very big place to try and find them. So
Brian's got the chat box.
we don't know for certainty for a lot of them, but I can say that a lot of the penguins we did see had time depth recorders and not the satellite tags. So I think the tags just fell off. They're not meant to be very invasive when they're just taped on. So it's just tape and a bit of glue to hold the seams together. It's very strong tape, but they're meant to not have a lasting impact on the animal's feathers. And you can see in a lot of the pictures that we had with the time depth recorder, you can see there's no disruption in the feathers above it, which was really great to see as someone who's kind of causing this impact to them. So we think that the tags mostly just fell off, or an antenna got broke, stuff like that. Yeah, the very young are fed basically by the penguins coming back in shifts. So unlike emperor penguins where they go to sea for weeks at a time, Gentoo penguins just rotate days in their breeding pairs. So the male will go off one day and then the female will go off the next day, and the other will stay and help keep the chicks warm or the eggs warm. And yeah, so they basically just feed their young what they caught earlier that day. They'll feed them when they come back with prey in their belly. They'll regurgitate a bit and feed it to their chicks. I don't know, but it was a workout. It was very difficult to chase after them, and we tripped in the diddle-dee. Like, it's not an even bush. Also, especially in April, they have berries that you can actually make into jam, and it's pretty good. We bought that in Stanley. I think I gave some to my grandparents when they came back. Yeah, so we were covered in berries, which are red, so it looked crazy on us. But yeah, so they are very, very speedy animals.
So the question comment, did Pingu influence you?
Mom and Dad, did I see Pengu when I was younger? They're nodding, so I was exposed to it. I don't quite have a memory of it, but it could be.
I'll throw up one last question. That critical time when they're in the molt and they've lost their waterproofing, how long does that take?
Yeah, so penguins take 2 to 3 weeks to molt all their feathers, and a lot of animals die during that time because they've spent a lot of energy trying to keep their chicks alive, and they get more demanding as they age. And then when they're finally able to support themselves, all the adults don't really get that long of a window to recover from that, to save up a lot of fat reserves to be able to molt all their feathers and regrow them. So they're really at their lowest body condition in April. And I can definitely say that there was a huge difference from when we were catching penguins in April when they were all tired to October when they were raring to breed and defend their nest. They were very, very aggressive. So with penguins, we don't really have to worry about their beaks so much. They can break skin, but it's like superficial. Their wings will do a lot of damage. They are very strong swimmers and they have a lot of muscles in their wings, and getting slapped with one of those, like I think it could probably break a finger. So we were very, very careful when we were handling them to be very cautious of their flippers.
Sir, I'm just going to invite Audrey up, so stay right where you are.
Wow, that was amazing. First of all, you totally blew us away with your incredible science, and the illustrated talk was just amazing too. The graphics and everything were done by an artist. We could tell that, but you helped us non-scientists really understand your work in progress. But then you're also a talented artist, and you just hop from one to the other like the penguins themselves. It was amazing to see. And your art is very powerful, and you're using it for a powerful purpose. So I wonder also if you will have your calendars available for sale after the meeting, when we, so that people might have a chance to buy them on the way out.
These calendars aren't mine. They're from Artists for Conservation. So Jeff sent us 10 to have for this event. So all of the proceeds from the calendar are going to the Owen Sound Field Naturalists, other than the 2 that we decided to raffle for fun, giveaways, door prizes. So I think there might still be some available through the Artists for Conservation website if these ones are gone.
So people could possibly order them in time for the new year.
Yes.
Yes. Okay, fantastic. And just in closing, I guess we're all super bursting with pride to have a local girl here on the stage who's done such a great job with both biology and art. And I would also like to mention, you know, the local connection and your former teacher, Jim Sebesta. I just wish that he was the one here today thanking you. He would have been so proud. So thank you very much, Sarah.